Now, we know that our life is not maintained by orphans, right? We are not alone, right? Even in the cell, the proteins don't work alone. Everything is coordinated, and it's just amazing and fascinating how proteins talk to each other and how they, I call this a relay system, right? In Olympics, you have the 4x100 or relay system. One has to give the relay to the other one has to do. So pretty much it's all a network, systems network, right? It's all about networks in our cell and in our lives as well. Now, so many social networks, there's nothing that is we can do without network. So we have organs, we have tissues. Tissues are made up of cells. Cells are made up of molecules, and molecules are made up of atoms, correct? So now a cell is an organization of millions of molecules. Proper communication between these molecules is actually extremely essential to the normal functioning of the cell. And to understand communication between molecules, we have to understand the arrangement of this particular atoms. In a cell, what happens is it's all molecular networks, right? So we actually have several different pathways, millions of different pathways. So what exactly happens in one pathway? If there is a problem, does it affect a pathway downstream? What do you all think? Actually, it actually does, right? So what happens here actually affects what happens here as well. That's what, again, we call it systems medicine, systems biology. It's all about a system as a whole, right? So we are all talking about all of those things. These are all the different biochemical pathways, and I'm sure that you've all actually seen all of these things. And a few different popular pathways that we all talk about is glycogen degradation, citric acid cycle, pentose pathway all of these different pathways. Why am I talking about all of these pathways? I'm talking about all of these pathways because at the end of the day, any pathway you take, right? This is the example of a citric acid cycle that's presented to you. This is a structural view of the citric acid cycle, right? So all the enzymes that are involved in the citric acid cycle, the structures are known, right? So why is that important? So if this structure is important, this particular enzyme is important because the product of this particular enzyme triggers this one, right? And the substrate, this becomes a substrate of this, and the product of this becomes a substrate of the next step. That's how all these cycles work, right? So if there is a problem here, then we already know that there is going to be a problem down here. A problem here is going to impact problem here, right? So that's what happens. So until we had all of these structures, we couldn't understand exactly why it's not binding, or it's binding too tightly, the products are not being formed, and maybe a hydrogen bond is lost, and all of those things, right? So looking at everything in 3 dimensions actually gives you a more insight into the functionality of all of these things, right? So that's where it comes to high resolution structural biology. So using atomic structure to understand how biomolecules communicate and functions, right? So what tools do we use? I'm showing a lot of pretty pictures. To my eyes, they all look pretty, right? They're all pretty pictures of helices and strands and all of those things that we're going to talk about. So what do you think are the tools that we actually use to make those pretty pictures? So this is X-ray crystallography. This is nuclear magnetic resonance. Electron microscopy, absolutely. Cryo-electron microscopy, yes. Spectroscopy, correct. Absolutely correct. So electron microscopy, you know what? Now even a lot of structures of COVID are actually done by electron microscopy. The reason is each one actually has pros and cons of using these techniques, but of course X-ray crystallography remains as the gold standard of methodology, and of course NMR as well, because I'm trained in X-ray crystallography. I think this is the best, but I'm just kidding. But a majority of the structures are solved by X-ray crystallography. We have nuclear magnetic resonance and electron microscopy. Also now, because we have lots of information, remember the big data we talked about, we have lots of information enough to do modeling, computation, right? Based on what we already have from all of these different techniques, and visualization and all of those things. So these are all the different tools that we use in structural biology, right, to solve the structures. X-ray crystallography, all of them actually give you the atomic resolution or the atomic positions, right? At the atomic level, they give you the coordinates of every single atom in your protein. Now, what are the advantages and disadvantages? Advantages of X-ray crystallography, it is actually, it's a gold standard technique, And it can actually, you can write, we were able to solve the whole structure of a ribosome using X-ray crystallography. You can imagine how huge a ribosome is, right? So there is no limitation on the size, right? NMR has limitations on the size of the protein that we actually can measure using NMR spectroscopy. Electron microscopy does not also have size. In fact, it's better than X-ray crystallography because you can actually look at the whole cell by electron microscopy. But what is different between electron microscopy and X-ray crystallography is the resolution of what you get out of electron microscopy is lower than what you get of X-ray crystallography. But I must say that, of course, a crystallography, there is radiation damage. And the biggest problem with X-ray crystallography is you need crystals before you have to make proteins as crystals. And I can tell you, having done that, it can be a nightmare to crystallize a protein. Not all proteins can be crystallized. So that is the bottleneck in X-ray crystallography. So each one has its advantages and disadvantages, and each one is actually complementary to the other. So NMR actually gives you hydrogen information because protons are the main center to nuclear magnetic resonance. X-ray crystallography, it's an electrons because it's X-ray beam. So electrons are the ones that diffract the X-rays. But then again, you can see hydrogens in crystallography. So we have to model it. Close your eyes for a second, just kidding, right? Don't sleep off. I just want you to imagine a protein sequence in front of your eyes, right? And just a plain old sequence, right? In front of your eyes. And now I want you to tell, if you have a structure, what can it tell that your plain old sequence, primary sequence, cannot tell you? What can structure tell you that a sequence cannot tell you? So it actually tells you how molecules are packed. It tells you interaction, right? And I think that Karina talked about polarities in a way. I think if that's what you were referring to, it actually interaction. And then it actually tells you how folding, how does they fold, which means which part of the molecule is close to which part of the molecule, right? Just imagine, right? This helix and this helix, right? In a primary sequence, say this is residue number 30 to 35, and this is about 500 to 550. I'm just making up these numbers. From a sequence, you will know that regions 30 to 35 is actually physically close in three-dimensional space to 500 and 550, right? 500 and 500, amino acid 500 is far away from amino acid 50 in a primary sequence. Would you all agree? But in 3 dimensions, because of the effect of folding, they come so close to each other, which we wouldn't know with just looking at a primary sequence. And of course, one of you actually told me about interaction of different parts. How does active sites and binding sites, Chandrani, I think, yes. So how does a ligand actually bind to, and where does it bind that are making the binding pocket? What are the regions that are exposed to the solvent? What are the regions, excuse me. And if you have 2 structures that are very similar, you can just superimpose them to figure out whether they're similar or not. And I think we're going to be doing this in our hands-on session when we do structures. We're going to be superimposing the molecule of hemoglobin, and we're going to a normal hemoglobin and a sickled hemoglobin, and we're going to find the difference, right? And then this is actually electrostatic and shape. And I think one of you actually asked me about how do you make, I don't know if it was Jeffrey, how do you make something specific, right? So this is how we look at it. We actually look at charge distribution. We actually look at hydrogen bond interactions. We actually look at all of these different interactions, and we look at the energies to figure out how we can make something specific. So we may actually change one of the atoms in the drug so that it changes its whole hydrophobicity and its polarity, or I'm just using those words that you guys have used. So we can actually change the interaction between this, and I'm sure although the structures are very similar between 2 things, we can actually make it very specific by altering the atoms in the drug to make it more basic or more acidic, or adding more hydrogen bonds, or or introducing a salt bridge or something like that, right? So that is pretty much how, it's a lot of work and a lot of things that go in. So I'm just giving you a simplistic answer of how something can be made specific. And then, of course, with a structure, you will understand what is a quaternary structure, right? that come together from two different polypeptide chains, which means those two polypeptide chains actually code for two different genes, right? So when two different things come together, and quaternary structure is made, or proteins that contain two different polypeptide chains coming from two different genes, those are the proteins that actually have a quaternary structure. And immediately you guys can give me an example of a protein that you all know, we've been talking about quite a bit in the last 2 days, is something that actually has a quaternary structure. Hemoglobin yes, hemoglobin actually has a quaternary structure. So we'll look at that. Then one of you actually said that we actually can do active sites and functional motifs. Yes, we actually can get active sites based on a three-dimensional structure. Okay, so now structure show and tell. Now you understand what structures can actually tell you, that you cannot get this information from just looking at just a sequence alone, right? What are the key players in this game? Proteins. Proteins play a very key role in our living system, right? They help in catalysis enzymes. They actually help in transport. There are transport proteins. They are information transfer proteins. We have hormones. So the whole protein world can be actually divided into, each one is given a job in the cell to do. And if they fail to do the job that they're given, that's when something goes wrong. When something goes wrong, and that manifests itself in, a disease of, for example, right? So everything has to be organized in the cell. Everything has to wake up at a certain time and has to let go of certain time. If they actually don't do, then that's a problem, right? So things happen at very femtoseconds inside the cell. So things have to go on and off, on and off. It's really fun to watch things in real life under a microscope, right? So these are all different examples of classes of different proteins. We have structural proteins, contractiles, and all of those things, right? You all know these are all different examples. Each one does a unique job. And some proteins, multitask. They are given more roles to play. So which means they're very, very busy, right? Some of us have lots of hats to wear, right? So major categories: regulatory proteins, they control gene expression; receptor proteins, they bind to things. You have structural proteins. Some of them, they just have to, fold in a certain form or a way to actually understand the functions of these things. Transfer proteins are proteins that transfer something from one thing to another. Do you all know an example of a transfer protein, something that transfers something from one to another? We have a lot of different transferases in our systems. Immunoproteins, these are very, very important proteins because they are the ones that are protecting us from foreign agents, right? So your immune system is very, very important, right? And then many more, right? I'm not doing justice to everything. This is just some examples, right? This is the world. So world of folds and functions. There are lots and different, each protein folds differently. And because fold is very closely related to its function. So each protein folds differently based on the work it's given. And do you think we have a finite number of folds in this world? Or you think that we have infinite number of folds or proteins? The other way to ask, membrane transport proteins. Yes, Jeffrey, that's correct. Now, do you think that proteins can fold in any way that they actually want to fold? In other words, do we have an infinite number? Yeah, microtubule bound proteins, correct. So do you think we have an infinite number of folds in this that proteins can have? The number of total folds we have, it can be 1 million, for example. No. Why? Yes, that is correct, Behul. Yes, we do have a finite number of folds. Yes, due to steric interaction. That's right. We have very finite number of folds. Do we know exactly what that number is? We still don't know the exact number, but there are numbers floating in the literature, like 10,000 folds and all of that. But my guess is that we're almost reaching the number of folds that we actually can see. Why? Because every structure that's coming out, new structure, is actually looking like something we already know, right? So the reason we have only finite number of folds is because the beauty is they all have to obey geometry. You cannot have any angle, any distance, anything that you want. You know what? At the end of the day, every protein has to follow the law of physics. So that's probably, physics is very important, and, everything follows rules. Now, do you all know that we have 21 amino acids? So why am I bringing this up? I'm sure you all know your amino acids, correct? The reason I'm bringing this up is each amino acid actually has an important role to play in a protein structure. Because each of them actually, have a different physiochemical property. So when they fold together, some of them coordinate and work well together, and others actually don't. So they group together so that they actually work well within the context of a fold, right? So each protein has a unique structure, right? And this is the amino acid sequence. And you fold this is what you get, right? So depending on where the amino acids are, they actually take sides where they want to be, right? So I'm sure that you know hydrophobic amino acids, hydrophilic amino acids, amphipathic amino acids. I just thought you all know all of that, and so I'm not actually going to be going over that. Just to tell you that hydrophobic means phobia, means fear of something, right? These proteins actually are scared of water, so most of the hydrophobic proteins are actually formed in the inner core. They actually stay away from water. Hydrophilic means these are amino acids that actually are like water, so they can interact well with water. So these are the amino acids that are actually formed on the surface of the protein. And then amphipathic means that these are amino acids that actually kind of have both properties of hydrophobic, and hydrophilic, right? So proteins actually are very, very organized. They're organized into primary structure, secondary structure, tertiary structure, and quaternary structures, right? And then protein structure has a hierarchical organization. So what is a primary structure? Do all proteins have a primary structure? What do you think? Does all proteins have to have a primary structure? Of course, yes, right? This is a no-brainer because we all start with the protein sequence, right? So everything has a sequence, and that tells you the arrangement of every single amino acids, right? So does every protein have a secondary structure? Yes, of course. Does every protein have a tertiary structure? Yes, of course, because the tertiary structure is the one that gives the fold of the protein, and that tells you how they interact with each other. Does every structure have a quaternary, every protein have a quaternary structure? We already discussed that. No, every protein does not have a quaternary structure, right? So the important things are the tertiary structures, right? How they interact. Now, I already told you, right? The sequence determines the three-dimensional structure. So everything that you want to know about a protein structure is already, the secret is already there in the primary structure, right? But the only problem is we are still facing a problem where we still don't know exactly how to get that information from ab initio using physics, first principles, right? And the next person who is going to solve this protein folding problem is where you have a sequence and you just fold it. You find a way to fold it from first principles, is the next Nobel Prize winning person in the field of structural biology. And I kind of have guesses of who that would be because I'm seeing a lot of work from that group. But that is where we're all waiting for, where you'll be able to fold a protein sequence. You just take a sequence and you give it to a machine and it's going to come up with all that. But of course, some of us will be out of a job after that, right? Because we're doing crystallography and NMR and all of those things, but first principles. But that's going to take some time because we're still trying to understand these proteins. Now, certain positions, how does a protein know how to start folding? What is the position of the amino acid that starts folding? So we have a lot of theories and hypotheses around this. Certain positions along the chain of the protein sequence actually act as a nucleation points. And chaperones, right? It's a type of a helper protein, and we know we have a lot of chaperone proteins that we have identified. And these are all the different proteins that actually help in assisting protein folding, right? They all have something that they assist in protein folding. Now, this is the primary structure, right? This is just a structure. And every protein, as you all know, has an N terminus, amino terminus, and a carboxy terminus. We always read the protein from the N terminus to the C terminus, right? That is our thing. And this is how it folds, right? So this is a primary structure. And the only thing that we actually could understand based on the primary structure is we actually may be able to say, okay, this protein could we have a disulfide branch, maybe forming a disulfide bond. But if there are 10 or 20 different cysteines, we don't know which cysteine is going to pair with which cysteine. And if the wrong cysteine pairs with the, there's a wrong pairing of these cysteines, what happens? You'll get a misfolded protein, right? So protein folding is very, very important.